Thanks for taking the time today to listen to Will, the newest wearable device leave you itching for more. This presentation is a collaboration between EAG and our toxicologist partner, Gradient. I am Mary Dothage of EAG and have had a lot of experience the past few years in working with these types of projects. I hope you find the information presented here both informative and interesting. Wearables are becoming more and more popular within the past few years. The definition of a wearable is any electronic device that can be worn on the body as an accessory. And with any device that comes in contact with a consumer, there are certain concerns and questions that may be asked. Some of the questions you may ask yourself include, are materials of construction suitable? Is a chemical allergen present? At what level is an allergen present? Is manufacturing of the end product consistent? Is a recall required? Essentially, is the product safe for consumers? If a product is found to be unsafe for the consumer, post-production, the loss of revenue for a company can be manifested in multiple ways. The loss of revenue can manifest itself in multiple ways, such as customer lawsuits, product recalls, and consumer product agency reprimands. And this actually happens. A quick search on the Consumer Product Safety Commission website or the CPSC website gives multiple examples of this. The first example of this is a glitter cell phone case. In this particular instance, the liquid in the glitter cell phone case leached onto the consumer skin and caused chemical burns. The second example is vegan boots. This particular product was recalled because of possible chemical exposure to benzidine. In the past, benzidine was a common precursor to dyes and pigments. However, its use has declined because studies have shown it to be a known carcinogen. 6,186 chemical substances are currently registered with the EU Chemical Agency. 20% of these compounds are classified as skin allergens or sensitizers. With numerous known allergens, how as suppliers of a product do we ensure consumer safety? Wearables are not a regulated industry, but we can look to other industries for guidance on how to complete this testing. One similar product would be medical devices. Both wearables and medical devices are similar, and both materials are in direct contact with the consumer and thus pose a similar threat in regards to safety. Medical devices are regulated and undergo strict testing protocols to ensure device safety. Wearables are not regulated and may not need to be tested as stringently. However, these types of studies are proactive measures that can be taken to minimize harm to the user of wearable products. A small investment during device development can save a large amount of revenue if a problem arises post-market. One way to assess device safety is through the use of extractable leachable studies. An extractable leachable study can identify compounds of concern that may come in contact with the skin during device use. Each extractable study should be designed with the device use in mind. So how do we go about designing a study like this? The truth is, it all starts with a conversation. Product information, including device composition and device use, are necessary to custom design an experiment. This ensures the study is representative of the device use. Samples will be analyzed to identify chemical compounds leached from the device. The identified compounds will then undergo a toxicological review to establish allergens present. The level of allergen will then be quantitated to finalize the risk assessment. Based on the risk assessment, a safety decision will be made. All the information is then gathered, and all that's obtained will then give us additional information about the product. There are many manufacturing steps to attain a finished product. Product manufacturing begins with raw materials, which are then molded into modular components. The modular components are then assembled into finished product. Many base polymers, plasticizers, UV stabilizers, pigments, and antimicrobial agents used during the manufacturing process are skin allergens. It is important to know that device composition for two reasons. One is to know what chemical compounds may be present so we can analyze for them. The second is to ensure the method of analysis is suitable to detect those compounds of interest. So another important aspect of study design is how the device is being used. Things like the duration of use and conditions of use are all taken into as factors of the study design. A device that's being used for a matter of hours may not be tested as rigorously as one that's being used continually. So you really want to customize your sample extractions based on the product use. This can also include solvent extractions and also the extraction time and temperature. We also want to know what components of the device are coming in contact with the consumer. It could be that there's components of the device that maybe don't come in contact with the consumer, and these may not need to be tested as stringently, or maybe not even tested at all. And then after we've discussed all this, we take one final look at the study design to make sure that it's consistent with established testing methods that have been established by ASTM, ISO, or USP. It's kind of a check just to make sure the testing is in line with what we want to present. Once the samples have been extracted, the extractions are then analyzed by analytical techniques, which can include GC mass spec, LC mass spec, ICP mass spec, and FTIR analysis. We use the data from the analytical techniques to identify chemical compounds within the extraction solution. This list will then be sent to a toxicologist for a risk assessment to identify any possible allergens. And with that, I'll turn the floor over to my colleague, Tom Lewandowski, to explain that process.
Well, thank you, Mary. So where do we get the data that we use to conduct risk assessment for skin allergies, right? So one possible place is from human studies, from patch tests that maybe some of you have experienced, where the chemical is put at a certain concentration on the skin, for example, here shown on the back, and we look for some sort of immune reaction, some sort of redness that might occur because of that. And that gives us an indication that the chemical could be an allergen and maybe what concentration is of concern. But human studies are fairly rare because, of course, conducting studies on people is somewhat ethically challenged. So we often get data from animal studies. And I've listed one here. It's called the local lymph node assay, where we paint the chemical on a mouse's ear and look then in the mouse's lymph nodes to see if there's an immune reaction that occurs because of that exposure. And that's a fairly standard study that can be done. We can also do studies in other animals like guinea pigs. In that case, the chemical may be injected or painted on the skin. And again, we look for some sort of indication that an allergy is occurring. However, in many cases, we don't have data from either human or animal studies. This is particularly the case when we have maybe unusual chemicals like breakdown products and other things that Mary might detect in her samples. And in that case, we have maybe other tools that we can bring to bear. There's, for example, read across, which is essentially looking through the process of analogy at similar chemicals with a similar structure that may also have data that we can then use to infer the properties of the chemical we're really concerned about. So it's really it's called read across, and it's basically reading across from chemicals with known data to chemicals where we don't have any data. A similar process really involves computational toxicology using computer programs, structure activity programs that really look at a chemical structure and use the power of computing and data that's been gathered over the years about how structures in the molecule may relate to particular forms of toxicity to make predictions for a chemical. So all of these things can be brought to bear to determine whether a chemical actually poses a hazard. And the questions we really look for are really just two questions. The first is the chemical a skin allergen, right? And that's a yes or no question. And if it is a chemical skin allergen, then the other question we have to answer, of course, is what level of exposure is okay, right? How much is safe? So once we have data from looking at the toxicity studies to understand whether we have a chemical hazard present, we then need to go ahead and take that information and put it through the process of risk assessment. And so what we do in a risk assessment for this particular concern is we first determine what the acceptable exposure would be, what the acceptable exposure limit would be, right, in micrograms per centimeter squared. And what we do there is basically take the data from the toxicity studies we just talked about and then apply some assessment factors, accounting for maybe areas of uncertainty. So Is the product used in a matrix that might enhance skin penetration? Is the product used in a way that occludes the skin or covers it that also enhances the skin penetration? How long might the product be used? Is it used every day, or is it used, once a year? These are all things that we can adjust for when we look at the assessment factors and determine the acceptable limit. That acceptable limit is then compared to the product exposure concentration, also in micrograms per centimeter squared. And that's determined from the lab result that Mary gives us, typically in micrograms, and then we divide that by the product contact area. And we're only really concerned about the surface area of the product that becomes in contact with the skin. And if the product has areas that aren't in contact with the skin, that's really not so relevant to the risk. Now, obviously, it should be fairly clear, we want that exposure value, the concentration the person is exposed to be less than the acceptable exposure limit, right? And if that is the case, if there is less exposure than the limit, then we know there is a minimal risk of some sort of adverse skin effect. And so the possible outcomes of this assessment, may be different types of things. We may actually have to make a go, no-go decision about maybe producing a product or putting a product on the market. We might actually want to document the risk assessment results as a way of having our internal stewardship records show that the product was looked at and determined to be safe. It's also actually really valuable to use this sort of process with an ongoing review of the product as it's being produced over time, maybe as production systems change, as you have new suppliers, to really make sure the product continues to be safe. So that can also be really valuable. So the types of studies we've outlined here, an extractables leachable study, can be really useful for identifying wearable safety issues and really ensuring the safety of the consumer. This type of study has real value early on in the product design process, where you might want to look at the potential safety of key materials that you would use in your product and maybe look for alternatives if there's a problem. It's also very valuable as a proactive measure to ensure consumer safety and thus avoid some of the recalls that Mary showed early on at the beginning of the talk, and also to avoid things like lawsuits. So a really worthwhile step to consider as you're developing a product.